Okay, thank you very much for the introduction. Just to say that I'll be also presenting on behalf of our research group. We are led by Prof. Van Wyk on the insect group. Part of the work that I will be presenting here is a collective work that was done in the group, just to surmise what we have accomplished up to this stage. Right. Okay, I must just go here. In the food industry, currently, it is widely accepted that by the year 2050, the planet Earth will be hosting 9 billion people. And to accommodate this number, the current food production will need to almost double. As you can see in the graph that is shown here, we see that the developing countries, most importantly, they would need to have an increase in terms of the food production or the food consumption per capita by that year, which is 2050. Currently, scientists are telling us that the oceans are overfished, and climate change and related water shortages could have profound implications on food production. Therefore, to meet this demand, we also have to take into cognizance the fact that 1 billion people currently on planet Earth, they are what can be categorized as chronologically hungry people. And how we process food products and what we eat and how we produce that of which we eat will need to be really evaluated. When we look into the African continent, we also know that food security remains a major challenge. And this is based on the context that globally, there are 2 billion people who are facing what we would term as protein malnutrition. So when we look at production of food practices and even the food that we consume, things like inefficiencies and food waste would therefore need to be reduced. So within this context, the need for new sources of nutrients therefore become evident. And so when we look at proteins, or when we think about proteins, proteins can be categorized into these three sources, where we have got animal proteins, plant proteins, and the recently alternative proteins, which will include things like lab-grown meat. So these are some of the sources that can be used to supplement the protein in our diets. But if we look at these alternative proteins, for example, the production of lab-grown meats, which has also received a lot of media attention in recent years, is still a little bit expensive for consumers. And when we look at plant-based proteins, there is a lot of hype currently around plant-based proteins. However, the limitation of these plant proteins is that they sometimes have what we term anti-nutritional factors, and some of them, they do not contain the complete amino acid profile. And of course, if we as food scientists look at the role of proteins in general, so it can be categorized into three forms. So we're looking at the nutritional aspects, we look at the physiological aspects, we look at the physical and technological aspects. And each of these aspects or these roles, they have distinct requirements, and they also look at different factors that can affect them. So if we talk about, for example, the nutritional properties of a protein and looking at the amino acids or the essential amino acids, these essential amino acids are indeed affected by the primary structure. So for a food processor or a food manufacturer, what we are mostly interested in to maneuver or to manipulate the protein is what we call the technological properties. And these technological properties are indeed influenced by the three-dimensional form that you have in the protein. So in South Africa, or in Africa, the practice of consuming insects, which is known as entomophagy, is not a new phenomenon. This has been practiced for over a decade. And recently, we've seen from 2019 going upwards that a number of researchers from the African continent have started working on edible insects. Our good friend, Dr. Robert Musungiri from Chino University in Zimbabwe is doing a lot of work on new product development using edible insects. And if we look at also work that is done by Dr. Kinyuru in Jomo Kenyatta University, also looks at incorporating edible insects into infant feeding schemes. So in Africa, researchers are already at hand working on edible insects. In South Africa, Dr. Leah Bessa, which some of us know, who's worked on the ice cream, which also received a lot of media attention, and they also started the pop-up store. And we also have seen media reports for Tito Mboweni, who was cooking using edible insects. So the recorded edible insects, they are close to 2,100 species that are widely consumed, and some of these includes mostly in the main 40% beetles. So edible insect fractions can be extracted from these edible insects, and these includes lipids, proteins, chitin. And if we look at the structure of these edible insects, of course, of proteins in general, we have the primary structure, the secondary structure, and the tertiary structure. But what is mostly important is the amino acids, which are the building blocks, because these are important for nutritional aspects. So in our research group, we worked with black soldier fly. Black soldier fly is an interesting fly that is being widely researched globally. It is because this fly, one of its advantages is that it does not feed. So it reduces the risk of carrying and spreading disease. It has also the ability to convert a wide variety of waste streams into quality proteins, fats, and minerals. It can also be read on cleaner waste streams, for example, vegetable waste or spent grain. So it can therefore find application in the food industry. But the question is, what are the key drivers in terms of using these edible insect proteins or these edible insects such as black soldier fly? So if we look at the consumer's perspective, consumers are interested in having products or food products that have clean label, less chemicals, less additives added to them. And there's also a growing interest in terms of sustainable and environmentally friendly sources that can be used in food production. There's also an increased traction, especially in Europe, of using insect-based and proteins and also healthy diets. And of course, another driver is the increasing incidences of allergenicity into proteins. But from a producer perspective, food producers or food processors are concerned about addressing the consumer demands for these alternative food proteins or products. And of course, if you're a food producer, we are also interested in finding a unique and competitive edge in the market. So what were our research aims? So our research aim here when we started the scientific exploration of this project was to firstly to study the effect of different extraction methods on the nutritional, technofunctional, and structural properties of black soldier fly larvae proteins, and also looking at the concentrates. And so what did we do? We started by designing a process from scratch. So we started looking at how can we treat these this black soldier fly. So we developed a a method to freeze dry and handle the black soldier fly. And from there, we went on to look at what can we use to defat the black soldier fly and remove the fat in order to obtain the protein. And we then looked at two techniques that seemed feasible. Sorry. So we looked at the alkaline and acid precipitation extraction method, and we also looked at the alkaline extraction method, just alkaline alone. And from there, we studied the nutritional and technofunctional properties. We looked at the scanning electron micrographs, and we used Fourier transform infrared spectroscopy to look at the fingerprints or the structural molecules that are present in these compounds. And then, of course, with the two protein concentrates that we obtained, we then looked at the zeta potential measurements of these proteins. And so the question is, what were the findings? So the findings that we firstly observed was that the alkaline and acid precipitation extraction method provided us with the highest protein content compared to only the alkaline method. And this alkaline extraction acid precipitation method gave us a protein content that is comparable to mealworm, which is around 74%, using the same kind of method. And then we also see that when we look at the technofunctional properties in terms of emulsion stability and capacity, this method was giving us the highest, I think it was around 90% to 100% in terms of emulsion stability and capacity. So if we look at just the emulsion capacity and stability, so the ideal qualities of an emulsifier for an oil in water emulsion is that it must indeed have a balanced amino acid composition. It must also have charged polar and apolar side chains. It must have or possess a good solubility in water, and it must also have a relatively stable conformation. So these properties are important for black soldier fly protein concentrates because we already know from literature that some of the plant proteins are generally recognized as less efficient emulsifiers, right? So here, these results are significant in the sense that black soldier fly, or as an edible insect, as we have obtained here, can then be potentially used as an emulsifier. So we also looked at the zeta potential for both protein extracts. We found the isoelectric point to be at around 4.5 for both extracts. But when we looked at the FTIR structures of these protein concentrates, we were able to observe the amide 1, 2, and 3 bands that are present there, indicating that these were indeed proteins that we had extracted using these methods. However, the intensity may be different depending on the method that you have then. So we also looked at solubility. So solubility is very key when it comes to functional properties or the functionality of proteins. And this solubility, if we would speak in general terms, we looked at the fact that solubility is largely dependent on the nature of the protein surfaces, which are normally in contact with the protein, and mostly in the food industry, that would be water. So if I would then generalize for you, I would say that a protein with a hydrophilic surface will be more soluble in water than a protein with a more hydrophobic surface. So if you look at the graph that we have here, we then see that based on this graph, if you look at the flowers, which would say the freeze-dried flour versus the defeated flour, so they are at the bottom, their solubility at low pH is very low compared to the protein concentrate. And of course, solubility being an important physiochemical and functional property of proteins depends on the hydration and the degree of hydrophobicity of these molecules that you have. So what we can then deduce from this graph here is the fact that the black soldier fly protein concentrates, they have a higher solubility at low and higher pH, and therefore they can then find also application in products such as beverages, which would be normally acidic. And then if you want to have something that has got a higher pH, which is around 8, you can still also have an application there. But between the two protein concentrates that we looked at using the IEP and the alkaline extraction, we see that the IEP at a higher pH has a higher solubility. And so with solubility, here we found that the minimum solubility was around pH 4, and then in the previous slide I've said the PI or the isoelectric point was around pH 4.5. So by the isoelectric point here, I mean, so it is that region where proteins have either their net charge is 0 because the positive and the negative charge, they cancel each other. And this what it does is that it favors the repulsion of molecules, and therefore it will increase the solubility of the protein. And previous work that has been conducted by other researchers in Russia, Onymensis, for example, it revealed that if you look at full fat and defeated flours, they also exhibited poor solubility. What then people do is that in literature, they will do an enzymatic hydrolysis using enzymes to break down the proteins. And some colleagues from Germany, they found that by doing that, they would increase the protein solubility up to 55%. So this work was then published this year in 2 publications. So the first publication by my research colleague Uzozo. So we found that she looked at the nutritional quality and the structural analysis of black soldier fly before and after defeating. And then the part 2 of the paper looked at the structural properties, the nutritional and the technofunctional properties of these flours and concentrates. And the second research aim, we then wanted to see what is the effect of conjugation time and temperature on the antioxidant and technofunctional properties of the black soldier fly protein. And so we look at the Maillard reaction as one of the important reactions that takes place in food products. Because if we want to further promote the application of black soldier fly proteins in the food industry, it is imperative that we understand the structural and the thermal properties, which will then influence the technofunctional behavior, such as solubility, such as emulsification, and indeed also looking at the antioxidant properties. So when we think of the Maillard reaction, we know that it is a reaction that takes place between proteins and sugars in food products. And of course, when we are going to have a product that has got these 2 components and there is application of heat, the Maillard reaction is therefore inevitable. In other words, it is definitely going to happen. So the Maillard reaction results in what we call products that are referred to as Maillard reaction products, or in some cases they are called conjugates. And these conjugates, some of them, they have been reported to possess antioxidant properties, enhanced solubility, and emulsifying properties. So then the question is, why do you go for this kind of a method? We know that mostly food manufacturers, they attempt by all means to avoid chemical agents in food production that may be potentially toxic and hazardous to the consumers. So the Maillard reaction is one of the modification methods of proteins that is generally regarded as safe. So that is why it's called GRAS. So how did we then design our experiments? So for preparing the conjugates, we used time and temperature combinations, and we kept our pH, our initial pH at 9, which was stable, and we looked at 50, 70, and 90 degrees Celsius, looking at 2 hour intervals. And for this first part, we looked at the antioxidant properties, the technofunctional properties, and we also looked at the antioxidant indices, which are Browning index, and also looking at pH reduction. So the findings here, we've seen that the DPPH, for example, and the ABTS radical scavenging activity, they increase with an increase in temperature. But if we zoom to the 90 degrees treatment, we see here that the emulsion capacity and stability was the highest between the 2 and 4 hours, and then it started to drop from 6, 8, and 10. And these conjugates, they also exhibited enhanced functionality, especially with forming properties. So this work was then also published last year in the journal, and so that concluded our second aim. So our third research aim, we then wanted to look at now, how can we characterize this native protein and the black soldier fly larvae conjugates using novel analytical techniques. So we used here FTIR, and we coupled that with chemometrics, which includes principal component analysis, and we also looked at SIMCA. But before we went even further, because we are inducing structural modifications in the protein, it is very important that the first step that you take is to look at the thermal stability. In this case, we looked at the thermal stability using the DSC. And so the DSC, what kind of information can we get there? So we can get information like the denaturation temperature of the proteins. And so here, using the example of the conjugates and the native proteins that is treated at 90 degrees Celsius, we can see some changes in terms of the denaturation temperature. And if you look at the conjugates, you can see that there is an increase in the thermal shift, or there's a thermal shift in terms of the denaturation temperature of the proteins. So generally speaking, we see that at 90 degrees Celsius, throughout from 2 hours to 10 hours of heating, there is no significant differences between the treatment at 90 degrees. But if you look at the comparison between the native and the conjugated protein, we see that there are some differences, and there's a approximately 16 degrees shift in thermal stability. This means that now the conjugates, they are more stable at high temperature. They'll be more stable at high temperatures compared to the native proteins. And this confirms what we know from literature, that after glycation, the tertiary and the quaternary structure of these food proteins, it is altered, and that will result in the functional properties being altered. And this confirms, again, the prior observations that we made in paper 3, which I just showed previously, where we observe these differences in the technofunctional properties. They are as a result of this thermal shift. Then we went further to look at another technique, which was the thermogravimetric analysis, which is TGA. So TGA is a tool that has been widely used in the food industry to monitor the mass or the weight of a substance as a function of time. So here in your y-axis, you've got the weight of the substance, and then on the x-axis, you've got the temperature. So if we select the temperature at 90 degrees Celsius as an example, we then begin to observe three stages of thermal decomposition that are taking place. So the first stage that is taking place can be attributed to the evaporation of the free and loosely bound water and other volatiles that may be present in the range of 160 degrees to 150 degrees Celsius. So the second stage of thermal decomposition, or the second stage that we see, is what we call is the decomposition or the degradation, which takes place at around 150 to 300 degrees Celsius. And if we would then compare what is happening or what we are observing in the native protein versus the conjugates, we see that most of the thermograms that are available or that are shown in the native proteins, they are closer together. So this signifies that the rate of decomposition is similar. And if we look at the conjugates, you can see that there's some differences, especially if you are looking at the conjugates that were heated from 8 hours to 10 hours. And then the last stage of decomposition that we see or that we observe is at the range between 400 to 550 degrees Celsius. And this stage is associated with the polypeptide decomposition in the native protein. And then again, there you see there's some differences between the native protein, which is the top part, which is A, versus B, which is the conjugates. And here we see that what we observe is that the lowest residual mass that was remaining after the heating process was around 10%. So we are confirmed, or we are confident that there are changes that are taking place in the native protein and also in the conjugated proteins when we are heating these up. So we then use an FTIR to build a model. So an FTIR is a non-destructive, precise, and consistent analytical tool that can be used to probe the structural modifications, especially in these protein sugar systems. This is because there are several readily identifiable regions in the mid-infrared spectrum where the technical fingerprints of the carbohydrates and the proteins do not overlap significantly. And what we see here, which is highlighted, is basically the carbohydrate region. And if you look at the graph or the spectra at the bottom here, it signifies glucose. And how do we know that this is glucose? Because you've got a high intensity in the carbohydrate range, and there is no intensity or band or whatsoever in the protein fingerprint region. And then if we overlay these thermograms and then or these spectra, you can see that the native protein, which is red, you then have the amine 1 and amine 2 bands. And so the carbohydrates, as you know, they range from 900 to 1,100, and their maximum at 1,028. So we already know that there are differences that are happening in terms of the structure, and this green analytical technique can then basically assist us to build a model where we have large sets of data. So these days we speak of what you call big data, right? So you would need to have a tool or a model that you can design in order to classify between the native and also the conjugated protein. And so we built this model. Yes, just about to conclude. So when we built this model, we could see that as you increase the rate of heating or the heating temperature, you start to have a separation between the native and the glycated proteins, and the loadings, they confirm which bands are mostly predominant or which correlate or can be attributed to these shifts. So when we looked at the soft independent class modeling analogy, it is based on 3 equations, so which is sensitivity, specificity, and precision. And then we were able to build a model that had an accuracy of 93%. So we then now are confident that the model works because we used a calibration data set and a test data set which had 60 and 30 samples respectively. And then when we looked at the thermograms, you can already see with the scanning electron micrographs, you can already see the differences between the glucose, the native protein, and if you compare that with the normal protein, which is widely used, casein, and where you can see that structurally these are different. So I will not go into detail with this. And then the work was also published last year. And then we've got 2 papers, and the paper by Zozo also looks at the amino acid profile, the changes as a result of glycation. So in conclusion, we see that the alkaline extraction with isoelectric point precipitation gives us a protein content which is higher and a high emulsion capacity. And of course, for future consideration, we should be looking at other green extraction techniques that we can use to extract these proteins, and perhaps look at consumer perception from an African or South African context. And if we want to add in more edible insects or conjugated proteins in food products, to look at legislations for human consumption. Thank you very much. There is a study that was conducted by Dr. Bessa on allergenicity of black soldier fly. They have not yet really designed much analysis. So when I spoke to the guys from FACTS, they are still working on the methodology to validate it. But there are some signs that there is very little reactivity to the black soldier fly protein. I think it's still going to be an area that will need to be researched for looking at also the other edible insects like mealworm. But to my knowledge, at the moment, it's only black soldier fly in the South African context that has been assessed for allergenicity.